over to you, Penny. Oh, and as she's muted, I forgot to say something in the announcement section, which is that next week is Michael Roderick, and the week after that we have a seminar which is Phil Cummins, and it's going to be on the Wednesday as part of the disaster series. And following that, we are proposing to have a short break, which we would normally have coinciding with the semester break, but actually in this case, we're going to do that associated with the time when, fingers crossed, we will be busy moving offices and taking all of our, well, maybe we will. But anyway, the 2 weeks of June, the first 2 weeks of June, we'll be taking time off. So,
fingers crossed.
Sorry, thank you. Penny, over to you.
Thank you, Louis. And thanks, Bree, for coming to our virtual seminar here today. It's a huge pleasure to welcome Bree into our offices at home. Bree is a lecturer at the University of Sydney. She received her PhD relatively recently in 2013. And in that time, she's done an amazing breadth of work. So she's worked on acid sulfate soils, on eutrophic estuarine sediments. She's looked at element cycling in those kinds of systems. And today she's going to tell us some of her really interesting work on dolomite in modern lakes. Bree, after her PhD, had a postdoc, and it was one of these prestigious OCE postdoctoral research associates at CSIRO in Victoria. And that was on looking at durable minerals for CO2 mineralization. So this is pulling CO2 out of the atmosphere and into minerals. And so that work then led on to an associate lecturer position at Monash, is my understanding, Bree?
Mm-hmm, yeah.
Yep. And then from 2017, so only 4 years out of her PhD, she's been a lecturer at the University of Sydney. So I think that that kind of career trajectory shows what kind of rising star Bree is. And I'm really looking forward to hearing her talk today, which is about biogeochemical drivers for modern dolomite formation. Thank you, Bree.
Thanks, Penny. Thanks for inviting me as well. It's a shame I can't come down, but hopefully I'll have the opportunity to come and see you all in person soon and have a look around the labs and that sort of thing. Yeah, so as Penny said, I'm a lecturer and I'm a research scientist at the University of Sydney. I'm with the School of Geosciences there, and I lead the geochemistry branch of the Geocoastal Research Group. And yeah, since we're neighbours now, I thought this was a pretty opportune time for me to first in this talk just kind of outline what my research interests are and the type of program that I'm running through the University of Sydney. And then I will dive a bit into this project I've been involved in for a couple of years now. It's mainly, we have a whole range of collaborators, but started this work with Sasha Wilson when she was at Monash University and continue it with her at the University of Alberta. We're looking at biogeochemical drivers that allow for dolomite to form under Earth surface conditions. So yeah, I just first want to give you a little bit of a background as to my, I guess, very broad ranging and I think probably continuously growing research interests. But I think it does come under this umbrella of process mapping and contaminant trapping. So I have expertise in aqueous geochemistry, chemical sedimentology, and mineralogy. As Penny mentioned, I did an X-ray diffraction postdoc at CSIRO in Victoria. And I use these skills to help me to interpret the biogeochemical signatures that are recorded in sediments. And these allow us to have a look at not only the natural processes that are occurring in sediments, but also allow us to look at how these natural processes might just be thrown out of whack by human interference, and ways in which we can fingerprint that. So ways in which we can look at a signature from a sediment and have an idea about the processes that are occurring, or have an idea about different sites that have been impacted by different activities from humans. So this has involved a lot of looking at really unraveling the dynamics behind sulfur, iron, carbon, and trace element cycles, both in natural systems, but also looking at those disturbed systems as well. Feeding into this has been looking at the impact of different land use change, looking at different land use decisions, and how this has influenced element cycling, but also looking at how climate drivers are influencing natural element cycling as well. And within that, looking at how these different processes are influencing contaminant mobility in landscapes. So my research also looks at different minerals, looking at their formation, transformation, and decomposition, and how that impacts the mobility of contaminants in a system. So for example, looking at how these processes impact the storage security of carbon dioxide within mineral hosts and how that might be compromised if conditions were to change, and looking as well at how metal contaminants move through landscapes in association with minerals when they either form or break down. Yeah, so I think what motivates these research interests is, the fundamental science. I'm just really interested in understanding different processes and how the earth functions, but there's also this applied perspective as well. I want to be able to feed this science into management and into different rehabilitation of landscapes that have been adversely impacted by people. So my team brings together different aspects of sediment chemistry and pore chemistry, surface water chemistry, and crystal chemistry to try and create, I guess, a complete look at a different landscape in its natural state and how that is altered by human activity. And I've just kind of presented a range of different photos from this variety of different field sites I've been looking at in my research. Today I'm going to talk about carbonate mineral formation, and this is a shot in the left-hand corner and the one behind me as well of the Purana Lakes in South Australia. This is one of our field sites that we're looking at for this project. I've been also involved in projects over in this corner here, looking at how environmental conditions are impacted by, for instance, disposal of dredge spoil on land versus disposal of dredge spoil in water, different land use management decisions that are going to have an impact on not only contaminant mobility, but those natural biogeochemical cycles and what they look like after humans have interfered with the natural function of the land. Penny mentioned I've done a lot of work in acid sulfate soils. This was my PhD research area. So I've got a great interest in looking at acidified landscapes and how that influences natural element cycling and contaminant mobility. And down here is a nice shot of some mangroves. I've done a lot of work as well, and we're just expanding a project looking at the impact of land use decisions and land use change on mangroves and the cycling of contaminants, how this impacts the sediments in the mangroves, but also how this impacts the adjacent systems as well. So today, the topic that I am going to be talking about, Dolomite formation, comes under this overall research theme that I have within my group, looking at unraveling the complex biogeochemical conditions that control carbonate mineral formation. And this is broadly, it's important for a variety of different reasons. Firstly, it provides us with the biogeochemical model for carbon dioxide sequestration. It allows us to have insights into how the earth really efficiently produces these high stability carbonate minerals. So it could potentially be information that is fed into carbon capture and storage technologies in the future. By looking at these processes, it also gives us an understanding of how carbonate sediments have acted as a sink and a source for carbon dioxide over geologic time. So how that has influenced carbon dioxide storage in the past and the potential influence this could have on future carbon dioxide stores as well. Carbonate mineral formation is really important in paleoceanography studies. And an example of that is the fact that people have looked at the precipitation of orthogenic dolomite to indicate changes in certain parameters like organic carbon burial regimes, surface water productivity, sedimentation rates, and the variations in the sulfate methane transition zone in sediments. And also orthogenic and biogenic carbonates are produced by life. So they act as really handy biomarkers in systems as well. So coming into that, the more specific research project I'm going to be talking about today is looking at unraveling the complex biogeochemical drivers that promote dolomite formation at the Earth's surface. So this is a topic that's fascinated scientists for centuries now. We know that if we look at the sedimentary rock record, dolomite is one of the most dominant minerals that we see throughout it. And we look at some deep sea sediments and see that dolomite is existing there. But nowadays in modern systems, the formation of dolomite is really rare. And so it's understanding what is driving the formation conditions for dolomite and how that has changed over time. And the modern dolomite that we see does exist in a handful of locations worldwide, and this includes the study site that we have in this project. So firstly, looking at the Purana Lakes in South Australia. This is a lake site that is precipitating dolomite and a range of other carbonate minerals that we are investigating formation mechanisms for, and also in the Caribou Plateau in Canada as well. There's a series of lakes there that precipitate dolomite and a range of other carbonate minerals. So looking at these lakes provides us with this really perfect opportunity to look at a natural laboratory for the precipitation of these minerals and try to unravel the underlying mechanisms that allow them to form. So before I go into the actual specifics of the project, I just wanted to give a little bit of a background on this mysterious mineral dolomite and why it is that we're looking at trying to understand how it forms, so I know that we're all on the same page with the research. So dolomite is a calcium magnesium carbonate mineral. It has 50-50 calcium to magnesium within its structure. And sorry, I'm just adjusting the screen. Yeah, so you have 50-50 calcium to magnesium within the structure, and that exists as alternating layers of calcium and magnesium cations interspaced by these carbonate ions. It's a really highly stable carbonate mineral, and sedimentary dolomite forms via a precursor mineral known as very high magnesium calcite, or VHMC, which is an orthogenic mineral. So that means that it precipitates as a chemical sediment in situ. And I've just put in a couple of X-ray diffraction patterns for both dolomite and very high magnesium calcite. And you can see that dolomite has these ordering reflections that are absent in the very high magnesium calcite pattern. So very high magnesium calcite, or VHMC, can be thought of as a really poorly ordered version of dolomite. Not only is dolomite seen really extensively in the sediment record, the sedimentary rock record, and not only is it really rare to see it in modern systems, it's also really, really difficult to precipitate in a laboratory setting. And that's because although it's thermodynamically favored to precipitate from seawater, it's actually kinetically inhibited in doing so for a number of reasons. So the magnesium ion, it has a very high hydration energy. It really wants to hold on to its water. And so preferentially, you're going to see precipitation of hydrated phases rather than anhydrous carbonate phases like dolomite. And also because there's low concentrations and low activity of carbonate ions in seawater as well. And despite centuries of research into this topic, there's still a lot of debate around the geochemical and microbial controls that facilitate the precipitation of dolomite in these low temperature systems where it's actually kinetically inhibited in forming. So one of the objectives of our project was to try and create the most comprehensive biogeochemical and mineralogical signature that exists for dolomitic sediments. And this would include a really detailed look at what's happening with the pore water chemistry, but also match this up with a really detailed look at the mineralogy of the system. And the idea here is that we basically create a fingerprint for these sites to better constrain the biogeochemical prerequisites for dolomite formation under these earth surface conditions. And also to allow us to understand how natural and dynamic redox cycles are driving dolomite synthesis. I feel like I've skipped the slide here now. Okay. Yeah, so we wanted to understand better how it is that dynamic redox cycles drive dolomite synthesis. What is known about dolomite precipitating environments in these low temperature settings is that it's a microbial process, and actually it's been really intimately linked with sulfate reduction. And we know that these microbial processes like sulfate reduction drive changes in sediment pore water composition, comparative to what you're going to see in the overlying surface waters. And this is something that's you're going to be able to see transitions with depth in sediment as you have changes to redox processes as you go down a profile. But beyond just looking at the influence of sulfate reduction on, different carbonate minerals precipitating in these environments, there hasn't really been a lot of research that investigates the more complex redox cycling and how this is going to influence the carbonate mineral assemblages that we see in carbonate playa systems that contain dolomite. So this is something that's actually pretty poorly constrained at the moment. And I think one of the reasons for this is that a lot of studies that look at pore waters for dolomite aren't particularly detailed. They don't tend to look at what's happening with the pore waters at great depth. It's just typically in the very surface of the sediments. And when they do look at anything beyond the surface, it's not, like I say, they don't cover a lot of different parameters. And besides from this, the mineralogy, while it's looked at in these sediments, is also not often particularly detailed. Having a quantitative mineralogy study for these systems is extremely rare. So what we wanted to do is to bring these two different data sets together to create a more comprehensive look at the geochemistry of dolomitic systems. And I think, the lack of this data up until now has been a real gap in our understanding the biogeochemistry of dolomite forming environments and understanding the processes that allow dolomite to form. Okay, so for the rest of my talk today, I'm actually going to be focusing on our Australian field site, the Purana Lakes in South Australia. But we do also have an international field site, the Caribou Plateau Lakes in British Columbia. And it's important to understand that we have, both of these lakes in here for a very specific reason. It's because there's just such vast contrast in the geochemical, lithological, topographic and climatic conditions between these different sites. Even though you're seeing really similar carbonate minerals precipitating at both, you're seeing reports of dolomite forming in both of these environments and other kinetically inhibited carbonate minerals such as magnesite. But yeah, when you actually look at the parameters present at both of them, they're completely different. So this gives us this opportunity to really uncover the underlying mechanisms allowing dolomite to form in these different systems. So if you look at the Purana Lakes here, it's a pretty mild environment. You're talking about temperatures that range between 16 degrees and 26 degrees. And, our mild temperatures at this site, we're talking the very low negative degrees. It's a hypersaline system. We've had a look at salinity with depth in the sediments, and it seems like it's about 7.5 times seawater in most sites. And the ions that come in from for carbonation in this system are coming from weathering of the calcareous dunes and coming in by the groundwater. Whereas if you can compare this to the Caribou Plateau, we're talking about really cold climates. Well, at least from my perspective, I grew up in Darwin, so this is pretty horrific to me. Average monthly temperatures between -15 and +13, and you know, we're looking at temperatures of negative 30 for their minimum. So it gets very cold in these environments, and you have the freezing of these lakes over winter and the thawing out over the summer, which is obviously not something that you see happening at the Coorong. There's also really variable salinity at these sites, and there's a variable sulfate availability across the lakes as a consequence of this, which is, you know, important from that perspective of dolomite formation being really intimately linked with sulfate availability. But what we're seeing is that different lakes that are forming dolomite in the Caribou Plateau, you can have tens to thousands of differences in the actual sulfate ion in ppm. So looking at, you know, the site itself is really interesting, but then being able to compare that to the variable conditions at the Coorong is, I think, going to give us lots of insights into what's happening here. And the ions from this system are coming in from weathering of the basalt. So it's a different landscape entirely. Okay, so I'm gonna, like I said, I'm gonna focus on Coorong. I'm assuming that my little bar up there is not blocking the screen. But yeah, so our field site in Australia is the Purana Lakes. It's this series of shallow hypersaline lakes that are situated along the coast of South Australia. It's about 200 kilometres southeast of Adelaide. And the three lakes that we chose to investigate in this study are Milne Lake, Pellet Lake, and North Stromatolite Lake. And these lakes were chosen on the basis that there have been reports of really vast contrasts in the carbonate mineralogy we're seeing across them. Despite the fact they're actually only a couple of kilometres apart, we, you know, really easily walked this entire circuit in around an hour, and that was stopping to look at lizards and that kind of fun activity. So it's actually really, they're really closely situated to each other. But yeah, but despite that, you see really different things happening in their sediments. North Stromatolite Lake, which we chose as our end member for this study, it hasn't been reported to actually have dolomite precipitating in its sediments at all. So it's like the non-dolomitic end member. Whereas if you look at Milne Lake and Pellet Lake, there has been reports of dolomite precipitating in the sediments here. Milne Lake in particular is really interesting because not only do you have the formation of dolomite, but you also have reports of magnesite forming. So this is interesting to us because although magnesite is less talked about in the literature, it is also kinetically inhibited from forming at a surface for the same types of reasons that you see dolomite not forming. So Milne Lake, by including this lake, it gives us that other extreme end member to the study on the other end of the spectrum from North Stromatolite Lake, where you have multiple minerals that are kinetically inhibited forming in the sediments there. So for us, it was really important to have, you know, all three of these lakes to include a really comprehensive look at the different spectrum of carbonate mineral assemblages you're seeing forming in these types of environments. The system is also ephemeral, which is really interesting, and it's actually something that's often overlooked. People don't seem to really have it in-depth, haven't really in-depth looked at what happens with the geochemistry of these systems as they dry out over the summer. And the drying out's pretty extreme. As you can see, this is us in the same, I think this was North Stromatolite Lake. So for us, it was important to capture these extremes in the seasonality of the sediment. We have taken sampling transects across the dry sediments through the summer sampling period, where we've, I think it was every 100 meters, took a core and sectioned it into 10 centimeter increments to look at the mineralogy of that system. But the data I'm presenting today is collected from our winter sampling. So when we collected intact sediment cores and extracted them to look at the sediment pore waters and also what was happening with the solid phase at those sites. So to create our biogeochemical fingerprint of the sediments in the winter sampling, we collected our intact cores. We section these into 2 centimetre or 5 centimetre depth increments, which were then really thoroughly homogenized. And then they were subsampled and either centrifuged for pore water extraction, and that was then filtered, and we measured a range of different analytes, and some of those were measured immediately if they were subject to change over time, or they were sensitive to oxygen. Some of those were preserved and taken back into the lab for analysis. And the same could be said for our solid phase. We sectioned that out, preserved it in different ways depending on how we were going to analyze it, and took that back into the lab. So at the moment, we're really kind of in the very beginning stages of, we've done a lot of data analysis, and now we're trying to pull together a story for it, and hoping to get something in a form that we can submit for publication by the end of the year. Today, I'm just going to step through some of the preliminary analysis we have, and I've actually been updating this morning because we've been meeting about the results that we're seeing coming from this system. So firstly, we saw some differences in the mineralogy of the sediments in the data that we collected for our samples as compared to what's previously been reported in the sediments at these sites. One of those differences is we actually couldn't find any bordering reflections in the dolomite. So this is via conventional X-ray diffraction, and there are other studies that have suggested there are ordering reflections that are captured using different types of technology, such as TEM. So our plan is to have a look a bit further into these samples and see if we can capture those ordering reflections as well. But I just wanted to make everyone aware of this because there's a lot of contentious, I guess, issues around the naming of dolomite versus VHMC. There are entire literature reviews written on this, and I'm not going to go into it in any detail here, except just to make you aware that at the moment, we haven't seen ordering reflections in our dolomite. We would have to class it as VHMC from that perspective. But in saying that, from a mechanistic perspective, we're not really that concerned. VHMC, or very high magnesium calcite, is another mineral phase that is very rarely seen in these low temperature environments, and it's suggested to be a precursor for dolomite formation. So by understanding its formation, we feel like that we're diving into what is allowing dolomite to precipitate in these types of environments anyway. We also saw some minor differences. We saw a bit of magnesium calcite at Nostromalite Lake that hadn't previously been reported. We saw that the magnesium calcite at Pellet Lake, maybe we had a little bit of that, but actually the main phases we were seeing was this dolomite or VHMC and also hydromagnesite. But the really interesting, I guess, carbonate mineral assemblages and how they changed with depth was we saw that at Milne Lake, particularly when you look down the profile and compare that with what we were seeing at Pellet Lake and Nostromalite Lake. So there was this massive variation in carbonate mineral assemblages at Milne Lake relative to what we were seeing at Pellet Lake and Nostromalite Lake. So this is our X-ray diffraction data for Nostromalite Lake. It is the data that we collected during that winter sampling period. If you haven't looked at X-ray diffraction data in this form, like a surface plot before, it's basically all we're looking at here is on the x-axis, we have degrees 2 theta, like you generally look at with an X-ray diffraction plot. On the y-axis, we have the depth in the sediment. So the 0 centimeters here is just at the sediment water interface. And basically what I've done is taken these X-ray diffraction patterns and stacked them together. And we're just viewing them from above. So anywhere you see an intense line, that's just reflection of a diffraction peak. So what we could see at Nostromalite Lake was that we had hydromagnesite, we had aragonite, and we had this magnesium calcite. But basically you saw these phases pretty much anywhere you looked in the sediment profile. And I have to bring the quantitative analysis into this to see exactly how much of the phases existed and how this changed with depth. But from a purely qualitative perspective, those carbonate mineral assemblages didn't change throughout the sediment profile, they're always there. The same kind of thing could be seen at Pellet Lake. You had hydromagnesite and dolomite existing throughout the sediment profile. And anywhere you looked at in that sediment profile, those phases were present. The story changed a bit when we got to Milne Lake, and you saw that there were the changes in the types of carbonate mineral assemblages you were seeing as you went down the depth profile of the sediment. And I'm just going to break this into zonation to make this a little bit easier to handle visually here. So firstly, the interesting thing that we noticed immediately was there was this kind of section in the middle of the sediment where you had dolomite and magnesite, these two kinetically inhibited minerals existing together. And this particular zone had an abundance of dolomite, and I'll bring the quantitative data up for this in a second. I just was able to get this morning. So, you know, both of these minerals existed, but this is a very dolomite dominated environment right in this middle section of our sediment core. Whereas if you look at the top of the sediment core, you actually have a system that's dominated by magnesite, and then you have less dolomite and some very minor magnesium calcite as well, which may be absorbed by other phases. We've been looking at the quantitative data this morning. Yeah, from a qualitative perspective, it did look like it was present. Then as you go down the sediment profile and you look at the bottom layer, you have the appearance of aragonite, and you also have dolomite and hydromagnesite, but you didn't see that magnesite layer so much. So this is the quantitative belt refinement data that I received this morning when I had a meeting with my collaborators, and it confirms what we're seeing in the qualitative data. You had these three very distinct mineralogical units in Milne Lake, where you had a dominance of magnesite in the top sediments, and then a dominance of this either dolomite or BHMC in the middle section of the sediment. So if you are to take this solid phase data by itself, it's pretty reasonable to start to think that maybe this middle section might present an opportunity to look at some niche biogeochemical and microbial conditions that could be pushing carbonate mineral formation towards BHMC or towards dolomite and favouring its formation over other stages, over other phases. And what's interesting is when we couple this with our pore water data, we see the same kind of zonation. So when we look at our redox, you can see we have our reducing conditions up into this 10 centimetre point, where you start to see it increasing into suboxic conditions, and that's where you see the dominance of dolomite taking over in this section of the sediment profile. And at the same time, you see this real dip in the pH of the system. So you're losing a pH unit in this section where dolomite becomes a dominant mineral. So we know that acidity, if it's produced in a system, is neutralized by less stable carbonate minerals. And I guess this just brings another layer of complexity to how we interpret this data, because it brings up the question of whether these carbonate mineral assemblages that we're seeing in the solid phase are purely reflecting the formation conditions for that mineral, or perhaps they're also giving us an insight into what phases might be remaining after other less stable carbonate phases break down with, for example, the introduction of acidity. This is something that we're not really sure of yet, but it's something that we have to take into consideration when we're interpreting our data. And it brings like this question of what is the role that mineral dissolution and precipitation, what role is it playing in this system? And is it something that is important in the formation of dolomite? This is reinforced when we look at our calcium and our strontium in the system, which you can see increases in these surface sediments here. And this is something that's not reflected in the sediment surface water. It's something that's not reflected in the magnesium profile of the pore water. and the magnesium more consistently follows what's happening with the salinity in the system. We do know that strontium substitutes in for calcium in the calcite structure. So this kind of begs to question whether we're having a calcium carbonate mineral that's breaking down in the top portion of our sediments that's producing this increase in calcium and strontium in the pore water. It's something that we have to investigate further, but it does bring us back to this idea about the role that mineral dissolution and reprecipitation is playing in these systems. And it also really highlights the need to include both pore water data, which is really sensitive to these mineral changes, along with your sediment solid phase data, which is something that's been missing in these systems in the past. Okay. And I just thought I'd really briefly mention this. This is entitled Upcoming Science. It's we have a DP proposal that has been submitted this year. Stuart Allen is actually one of the CIs on this, looking at how dolomite formation is influenced by these more complex redox processes that are occurring in the sediment beyond just looking at sulfate reduction. And one of the components of this is not only looking at how dolomite links to sulfate reduction in the sediments, but also looking at what happens when the reverse is seen. You see a flip in the redox conditions and you have those sulfide minerals that precipitate during sulfate reduction and that coexist with those carbonate minerals breaking down because you have oxygen introduced into the sediments due to drying processes. So this is something that has been, I mean, it's been considered, but not really investigated thoroughly in these sediments. You haven't previously seen these sediments looked at for sulfur speciation, and that's something that we want to do. And also look at how these redox-induced changes to sulfur cycling are driving potential formation of dolomite in these systems, or how at least they're contributing to changes in carbonate mineral assemblages. So to do that, I actually have some data that I collected from the Tenzing beamline in Taiwan, looking at sulfur pH X-ray absorption analysis, trying to look at the speciation of the solid phase and what types of sulfur species we're seeing with depth. And we're going to couple that with selective extractions to reinforce that data. And yeah, this is just the point of looking at this connection between carbonate minerals and sulfur speciation in the sediments, a really important one due to these ephemeral cycles. So we want to couple this investigation of sulfur speciation and carbonate mineralogy with looking at field and laboratory incubation experiments, and also combining it with our field summer sampling, which is, you know, those samples are also in the process of being analysed at the moment. Okay, and that's all I have time to show you today. Yeah, hopefully next time we meet, I can give you a little bit more in-depth update on where we are with that data. Thank you.
Thank you very much. It's very hard to tell that people are clapping furiously from behind.
Yeah.
It's a very strange experience talking to my screen. I got used to it with lectures, but yeah, a little bit weirder with seminars.
Yeah, we try to do this clapping symbol, but it doesn't always happen. Everyone's always. Thank you, Bree. That was a.
I felt the claps. Thank you.
Does anyone have a question for Bree on this work? Yes, Brad.
Well, I guess I have to, almost have to ask a question, given I work on dolomite.
Oh.
Yeah, it's been fun because we've been working on dolomite in tropical systems.
Oh,
I suppose Mirinda would say, well, it's not just tropical systems anymore. She's grabbing crustose coralline algae and coralline algae from all different latitudes all around the world and finding dolomite in them.
Yeah, I'm really interested in that research. Actually, I read a little about.
Yeah. So one of the things, certainly on the reef, what's consistent with what you've shown here, and thank you for the talk, by the way. I thought you did
Not
great.
a problem.
Certainly when you're on the algal flats, some of the things, we have done some pore water studies, and it certainly does go reducing in a real hurry. So we do know that it is reduced, but we really haven't really paired up sulfate reduction with the dolomite formation within the crustose coralline algae. Certainly there are places where it is reduced and places where it isn't, because there are places where you see it, basically just hanging out. But in terms of the microchemistry within the algae, it's hard to know what the redox conditions are. But I was curious as to what you think the physical mechanism might be in terms of what is, can you put your finger on something? What is the redox driver for freeing up that dolomite formation?
Oh, well, from a physical perspective,
just a physical chemical perspective.
Yeah. I mean, it's the redox driver is definitely these microbial processes in the sediments, and that's been pretty comprehensively shown. That's what we're interested now in expanding on that current knowledge is, people look at this from the perspective of sulfate reduction. Rightly so. I mean, the sulfate reduction in this system is going to be important, but I don't think that it is the key or the only thing that is influencing processes in sediment, and I think that's largely been ignored in the past. So yeah, I think it's going to be a combination of different reductive processes when we really tease apart what the data is doing. But yeah.
I suppose the second question I have is, do you have a feeling for what kinds of algae you've got mixed up within your lake sediments?
Oh, yes. Well, we do have micro algae itself. We do have that. We have the biology components of the collaboration on that. I couldn't tell you exactly what it is, but we have also done some microbial assemblages in these as well. And once again, I couldn't tell you the specifics of that. But yeah, I'd be interested in talking to you more about the systems that you guys are looking at. There are microprobes that you could potentially put into your algae to look at redox conditions. Yeah, I don't have a great understanding of what types of what your system looks like, I guess.
Okay. Thanks again, Bree.
No worries.
Any other questions? I have one, Bree. Do you so the data that you've shown is pore water and mineralogy. Do you have any data showing changes day night or showing changes on the small scale?
Like diurnal,
localized.
Yeah, no, we don't. It would be interesting to look at for sure. Yeah, we haven't gone there yet. Yeah.
Any thoughts for doing experiments to simulate some of these processes?
Yeah, for sure. I mean, we're devising laboratory experiments for trying to, I guess, recreate what we're seeing in the field and seeing what comes out of it in terms of mineral precipitation. We haven't discussed diurnal, but that's interesting. It's something that you could potentially do in a laboratory setting. Yeah, so the answer to your question is yes, we have laboratory experiments that we have planned, but we haven't started them yet.
Cool. And well, I've got the microphone. So you started at the beginning talking about CO2 sequestration, And I'm wondering how you're sort of thinking this project will link back to that question, and also link into sort of transport of the water and
Mhm.
wetting and drying over time within these lakes. Can you sort of comment on the best way to sequester carbon dioxide out of the atmosphere in a lake like this?
Oh, yeah, a tricky question. I guess the purpose of these studies is a mechanistic look at, the way, if we want to sequester carbon dioxide in the most efficient way, we would copy the way that nature does it, because not only in these systems are we seeing these really, really highly stable carbonate minerals forming pretty fast compared to, what we've been able to simulate in the laboratory, and in much greater concentrations, much greater quantities. So yeah, I guess it's just a mechanistic, it's trying to figure out how nature does this, because if we could do that, then it has the potential to be fed into carbon capture technologies where they're trying to produce highly stable carbonate minerals for carbon capture. At the moment we're looking at minerals that are a lot less stable than magnesite and dolomite, but yeah, the dream would be if you could produce those higher stability minerals in a less energy intensive way than they are already. Yeah.
Cool, thanks. I don't see any other hands up. Does anyone else have a question? Brad, your hand's up. Is that just still up from before, or?
Yeah, that's just a.
It's waving.
I don't even know how to take it off.
Oh, you just press the hand again. It's an on-off hand.
Oh, okay.
I can jump in, Penny. It's Patrice here.
Hey, Patrice, go ahead.
Yes, Bree, thanks very much. Very interesting talk. I was wondering if you or some of your colleagues are looking into the DNA analysis or the genome of the microorganisms that live in those sediments.
Yes, yeah. So we collected some samples for that. Emma Gagan and Gord Southam at UQ are looking at the DNA and the microbial groups living in the sediments.
Mm-hmm.
I don't have that information, unfortunately, but it is something that we're looking into as well.
Right, thank you.
Sometimes it takes people a while to unmute.
Yeah. Me and every lecture I've given this semester.
Any more questions? Stuart, what's your involvement? What's Stuart's involvement, Bree?
He's dating for us.
Yeah,
so he's been doing the dating of the sediments, and the isotopic signatures for this DP that we've proposed for laboratory samples, and hopefully for some lab samples as well. So you can not only look at dates but also confirm the carbon source going into the minerals, whether it's atmospheric or not. Yeah. That's what Stuart would have said. I'm sure he hasn't bailed early. He must be there somewhere.
Yeah. I have another question about the results. You showed us the carbonate minerals.
Mm-hmm.
Do they make up a lot? How much of the material is actually amorphous? Do you quantify the amorphous content, and is that important in the whole carbon story and capturing stuff?
Yeah, that's a really good question. From the X-ray diffraction analysis, it's not easy to quantify amorphous. I mean, there are ways in which you can kind of assign it a percentage based on the other minerals. But yeah, it's not something that's particularly easy to quantify.
So you're not putting in like a tracer spike?
Yeah, no, we have.
Oh, you have? Okay.
Yeah, we have. But yeah, it's I think that yeah, there's only I don't think it's going to be entirely possible to see amorphous versus crystalline in terms of what you could, basically what I'm trying to say is you can tease out how much of the phase is crystalline versus how much is amorphous, but it seems like there is a lot more to the amorphous story than has previously been thought in carbonate systems. And you can have a range of different amorphous phases in the same system that might actually contribute to carbonate mineral formation in different ways. And that's actually something that Sasha's been working on at the University of Alberta with some of her postdocs. But yeah, so she's getting some really interesting results out of that, you know, the amorphous content in these systems actually does seem to be quite important. And I don't think you just can't fully capture any complexities of that with X-ray diffraction. So yeah, one to look at in the future as well.
Yeah, have to come and do some IR on your samples.
Yeah, definitely. That sounds fun.
Yeah, it would help out, I think.
Yeah, awesome.
Yeah, cool. Well, I think we've lost a lot of our participants. And so that's usually a sign that she'll wind down.
Time to go.
Time to go. So yeah, another round of applause from me. Thank you very much for
Thanks, Penny.
Interesting talk, Bree.
Okay, thank you.
Okay.
Thanks for having me along.
Yep. Bye.
See ya.
I usually go last, Bree.
I'm trying to leave. I don't know how.
I usually hang out and say good job to the speaker while everyone.
Okay, I'll wait with you.
I don't like turning the lights out because people go, oh, I just got dumped out of the show.
You can usually tell who's left at the desk and is off, like making a sandwich or something. Around after the seminar's over. That's what I tell my students anyway.
Yeah, yeah.
Actually, it's funny. One of my tutors said the other day that he asked a question in his tutorial trying to like prompt some discussion, and he saw about 80% of his students just leave immediately. So he's wiser. Zoom. How dare he?
That's hilarious.
Yeah.
Well, I know Louis actually left, even though we can see him still, because he had to go pick up his son from daycare.
Yeah. No,
so he sent an apology privately.
Oh yeah,
to me to tell you that he apologizes.
I completely understand. Yeah.
Yeah. Yeah. Anyway, that's really interesting work. I enjoyed that.
Yeah, it is really interesting. I feel a little bit like because we're kind of just all meeting and trying to tease apart the story now, and I think it's maybe a couple of weeks off being ripe, like really ripe. But yeah, no, it's kind of coming together.
Yeah.
Yeah. We're still recording.
So with the IR, you could look at the minerals. Oh, are we? Oh, okay. I just turned.
Yeah, I don't know if there's a way to turn that off.
There is. I just need to get back into that screen.
Depends, I guess, if you were the person that turned it on in the first place.